grade 12 learners. Welcome to today's Life Sciences lesson. My name is Agnes Moabelo. This lesson is brought to you by the Gauteng Department of Education in collaboration with Saibono Discovery Center. We've got a WhatsApp number there where you can text us your messages. We don't need videos. We don't need voice notes. You can only text your questions. The number is as follows: 079-454-9535. I'm going to repeat the number: 079-454-9535. This is only for your text messages. All right. So today's topic is brought to you just in time by myself, Agnes, and my co-presenter, Samantha. So the topic for today that we're going to be looking at is meiosis. We're only going to do part 1, and part 2 you will do it the following week, right? Now, meiosis takes 21 marks of our paper 2. Right, according to exam guideline, with meiosis, you need to understand the following. You need to be able to discuss different stages of meiosis. That is why, as we'll be continuing with this topic, we'll be showing you those pictures, we'll be showing you those stages whereby you are going to quickly differentiate between the two phases, that is meiosis 1 and meiosis 2. Again, you need to be able to compare mitosis and meiosis, right? Also, you should be able to differentiate between meiosis 1 and meiosis 2, right? And then at the end of the topic, you should be able to discuss non-disjunction and the Down syndrome. So if you know all of these items, you are able to score your 21 marks. I'm going to encourage you that you need to start working very, very hard. I know right now you are busy preparing for your term 1 test, you are preparing for your term 1 practical, and obviously you need all these topics for the term 1. Right, you can ask yourself, where does meiosis take place? So meiosis takes place in plants and animals, right? So in animals, we know meiosis takes place in the testes of the males, okay? And then in females, it takes place in the ovaries of the females, okay? Now with these structures of the ovary, the testes, we are going to learn more about them when we do reproduction. For this lesson now, you only have to know where meiosis takes place in animals, in the testes of the males and in the ovaries of the females. Right? Also, meiosis takes place in plants. Now, in plants, where exactly? Right? We've got the female part of the plants, which is the ovaries. We've got male part of the plant, which is called the anther. Now, if we were to look at the ovary, what makes up the ovary? This is also important. The ovary is made up of stigma. This is the stigma here, the style, this area, and also the ovary. So the 3 parts forms the ovary. Stigma, style, and ovary, they form the pistil, right? So meiosis takes place in the ovaries. Now, when we look at the male part, with the male part, we've got the stamen, and with the stamen, we are looking at the anther, this structure here, and the filament, okay? Now, inside the anther, we have the pollen. This you can see is the pollen, okay? Now, in conclusion, in the place of where we find or where meiosis take place, we're saying it takes place in the plants and animals, it takes place in the ovaries and testes of the female, and males respectively. And then in plants, it takes place in the ovary and also in the anther. Yes, you have learned about the structure of the flower in your grade 11. You know this part, the bright color is the petal, then we have the sepals here. So for now, you need to know where does it take place. Anther and ovaries. Right? Now, you also need to know mitosis, because later on we'll be comparing mitosis and meiosis, right? Now, what is meiosis? Mitosis is also one example of cell division, right? It's a process where we can observe the cell dividing. So, this is the way whereby you can easily identify those phases, all right? Now, all in all, let's look at these phases of mitosis. We've got the interphase. Okay, the first one is the interphase, which is the preparation phase. Remember, in the interphase, that's where you find the chromatin network. Okay, now during the mitosis, they will form the chromosomes. Then chromosomes start to be visible in the prophase. Okay, let's look at those stages. And this format will help you to understand and master the phases, right? We've got IPMET. How does it help you? I for interphase, this is the first one. P for prophase, right? M for metaphase, A for anaphase, T for telophase. This is IPMET. So actually, what is going to happen if you draw those phases and look at what is within and what is changing, that will help you to understand each phase. So chromosomes starts to be visible in prophase, right? And then they are going to line at the equator, where in metaphase, can you see that? This is the difference, and we have in the formation of the spindle fibers there. Then during the anaphase, what is going to happen? These chromatids are going to be pulled to the opposite poles. Then cytokinesis, division of the cell, of the cytoplasm. So if you can check, the cell is ingesting at this area. So this means we're having cytokinesis taking place, in resulting to two daughter cells. And if you compare, the number of chromosomes that is within differs with the number, no, they look the same as the numbers that we had originally. Now, what is mitosis? A type of nuclear division that produces identical daughter nuclei, each having the same number of chromosomes as the parent nucleus. They have the same number of chromosomes as the parent nucleus, right? Then let's compare it to meiosis. What happens during meiosis, right? It is a cell division whereby a diploid cell, this is a diploid cell, undergoes two cell divisions. Let's look at those two cell divisions. It's meiosis 1, then we have meiosis 2 here. So for meiosis to take place, a cell is going to divide, whereby a diploid cell undergoes two cell divisions. Like I have said, these are the two cell divisions, and they divide to form 4 different haploid cells, or the gametes. Now let's look at this diagram and understand it better. So this is a diploid cell. Now DNA replication takes place. Remember when we were doing DNA code of life, we have introduced DNA replication, whereby DNA makes a copy of itself. So here we have got a haploid cell dividing into a diploid, replicating into a diploid cell. Now what will happen to that diploid cell? It undergoes meiosis 1. Then by undergoing meiosis 1, it is going to result into 2 haploid cells. Now those 2 haploid cells are going to undergo meiosis 2. Remember, meiosis is a cell division whereby a diploid cell undergoes 2 cell divisions. Okay, this diploid cell that has replicated from the original will undergo meiosis 1 and result into 2 haploid cell. Now the 2 haploid cell, they will undergo meiosis 2. Now when they undergo meiosis 2, each haploid cell will divide into 2 haploid cells. This one also divide into 2 haploid cells. At the end of meiosis, when it has taken place, we are going to have a result of the formation of 4 haploid cells. Hence, we are saying it undergoes 2 cell divisions and divide to form 4 different haploid cells. These are different haploid cells. So what is important? The diploid cell undergoes 2 meiosis division, that is meiosis 1 and meiosis 2, right? Then you need to also learn, because you'll get these terms in your exams, you need to differentiate between a haploid and a diploid. So the way we're writing a haploid, we write it using the small letter n, that will be a haploid, and then a diploid is 2n. Okay, now every species has a specific number of chromosomes in their nucleus. Okay, just before we move on, if you look at this diagram here, so this shows a haploid sperm cell. It's a haploid sperm cell. Okay, let me write it. Haploid sperm cell fusing with a haploid. All right, and there. Okay, a haploid sperm cell fusing with a haploid ova, they will result into what? A diploid zygote. All right, so I want you to understand the difference between a haploid and a diploid. So what we say now, every species has a specific number of chromosomes in their nucleus, right? Now, what is the difference between haploid and diploid? I gave you the symbol that you can use to write, to represent those. So having just one set of chromosomes, that is, chromosomes are unpaired. One set of chromosomes, that is haploid, and then having double set of chromosomes, chromosomes are paired, right? Other differences is that sex cells, the gametes, sperm cell or the ova, they have got 23 chromosomes. Please remember that one. There's gametes, that is the sperm cell and the ova, they've got 23 chromosomes. Hence, if the sperm cells and the ova, if they fertilize, what are we going to have? Look at this one when we compare the two. The somatic cells, which are the body cells, they have 23 pairs of chromosomes. And then 23 pairs will give us 46 chromosomes. This means in the somatic cells, we have 46 chromosomes, whereas in our gametes, which are the sex cells, they have got 23 chromosomes. So do not get confused, because once you say 23 chromosomes, you're going to get it wrong. It should be pairs when we talk about the somatic cells, right? So if we talk about the whole chromosomes, they are 46. And then in pairs is 23 pairs, which brings that number to 46 chromosomes. Okay? And then this is what I've just mentioned in terms of the haploid and a haploid giving rise to a diploid number of chromosomes. So when a male, which is the gamete is haploid, and when a male fuses with a female, right? When we talk about a female gamete, it will be haploid. A male gamete, it will be haploid. When they fuse together, what is going to happen? We're going to have a zygote that is diploid. So N fusing with N will result in 2N. Haploid fusing with haploid, they will give us a diploid. So a male gamete fusing with a female gamete, they will give us a diploid zygote. Right, we've spoken about those phases, the IP met, right? Now with the interphase, remember we have said it is the preparation phase. Okay, so what happens during interphase? DNA replication occurs. It makes a copy of itself. Then each chromosome will now consist of two chromatids, Right, this is an example of a chromosome. Okay, this is a chromatid and a chromatid. Two chromatids forms a chromosome, right? These are homologous chromosomes. Each is a chromosome, right? Paired together, they are homologous chromosomes. Do not forget, a chromosome is made up of two chromatids joined together by a centromere. All right? So, DNA replication will occur and chromosomes will now consist of what? Two chromatids joined together by a centromere. So it's important also to know how to draw and label the structure of a chromosome. That you can be asked. Then DNA replication helps to double genetic material. So DNA replication helps to double genetic material so that it is shared equally during the division. Right, we talk about single thread chromosome as it appears in a cell before DNA replication takes place. Right, what is a single thread chromosome? Now, a single thread chromosome, it is a chromosome that appears in a cell before replication takes place. So before replication takes place, you talk about single thread chromosome. Right, now, after replication has taken place, you can talk about the double thread chromosome. So you can ask yourself, what are double thread chromosomes, or what is a double thread chromosome? A double thread chromosome is a chromosome with two chromatids after DNA replication, right? This is an example. Before replication, you talk about a single thread chromosome, thread chromosome. That is before replication. When DNA replication takes place, when replication takes place, the results we're going to have what? Double thread chromosome. Chromosomes with two chromatids. Remember, chromosome with two chromatids joined together by a centromere. Then you can talk about the double thread chromosome. Okay, how do you differentiate between the two? A single thread chromosome is the one that you get just before replication take place, and a double thread chromosome, you get that chromosome after DNA replication has taken place. And with double thread chromosome, think of what? Think of chromatids joined together by centromere to form what? To form a chromosome, right? And also, you need to talk about these homologous chromosomes. Don't just refer to them as chromosomes. So maternal, paternal, we refer to them as homologous chromosomes, right? Now, homologous pair of chromosomes, yes, chromosomes that are identical. Those are chromosomes that are identical. Now, they are identical in shape. Look at this one, right? Look at these chromosomes. Look at these two chromosomes. So I've made them clear to you so that even with color, you can see that they differ. One is from maternal, one is from paternal, right? one will be from maternal and the other one will be from paternal. So what we're saying, the homologous pair of chromosomes should be identical, or they are identical. How are they identical? They are identical in shape. They've got the same shape. Look at this chromatid. They've got the same shape. They've got the same length. Look at their length here. Same length, right? And also, they've got the position of the genes or alleles. Position of the genes, look at the position of the genes here. The position of the gene is the same. The length is the same. The shape also is the same, right? Genes coding for same characteristics. So chromosomes that are identical, they also have genes coding for what? For same characteristics. And then we also have location of the centromere. Remember, with location of the centromere, that's the area, that's the part where it connects the two. It connects the two chromatids, right? This is the centromere. This is the centromere. That is very, very important again. Right. There are two chromosomes of each kind, one from mother and one from the father. One from the mother is maternal, it comes from the mother. Paternal, it comes from the father. Okay, we have spoken about the single thread versus the double thread chromosome. We have mentioned in the previous slide that the single thread, we're looking at what? At the chromatid there. It's a single thread chromosome. That is, we can see that before replication takes place. You can talk about what? A single thread chromosome. Look, after replication has taken place here, then we're going to have the double thread chromosome. So these are the double thread chromosome. Now the double thread chromosome, if you look at it, it gives us what? A chromosome. Before replication takes place, we've got a single thread. You can refer to it as what? As a chromatid, all right? So before replication takes place, talk about the single thread chromosome. After replication has taken place, it leads to what? You can talk about double thread chromosomes. Double thread chromosomes are made up of what? Two chromatids joined together at the area called the centromere. So that is why I've said to you, you need to know how to draw and label the structure of a double thread chromosome, of a chromosome. So what makes the two differ? Single thread, that is before replication. After replication has taken place, you will have double thread chromosome. Okay, for every topic, girls and boys, you need to know the terminology. You need to know terms that are related to the concept there. Now we've got a terminology that you need to know. We've got single thread. We've spoken about that one. Is unreplicated, right? If it happens that the examiner asks you about the replicated, differences between replicated and unreplicated chromosome, take it back to the explanation that we did in our previous slide, that a single thread is happening, or you can get that before replication take place. Double thread is replicated chromosome, that you can talk about them after replication has taken place. So single thread refers to chromosomes and it appears before DNA replication. So this is actually a summary of what we were just discussing. Double thread, which is the replicated chromosome, girls and boys, it refers to the chromosomes as it appears after DNA has taken place. Remember, a chromatid replicating itself, we're going to have two chromatids that will give us a chromosome. So you can talk about replicated because that chromatid make a copy of itself. Then we ended up having two chromatids forming what? A chromosome. And the part that joins the two is what? is the centromere. So replicated, double thread, unreplicated, single thread chromosome. Looking at the chromatid, it refers to each of the two threads of a replicated chromosome, right? Before replication takes place, we've got what? A single thread. Now replication takes place, it makes a copy of itself. Then we talk about what? A double thread chromosome. So to get a chromatid, when we talk about a chromatid, know that with a chromatid, you can talk about it. That is before replication takes place. But immediately when replication has taken place, then you can talk about two chromatids, because it has doubled, two chromatids forming what? A chromosome. Then let's talk about the daughter chromosome. Remember, we've got single thread, double thread, chromatid, then daughter chromosome. What is a daughter chromosome? We refer to each chromatid after it splits from its sister chromatid during Anaphase 2. You will see that when we do those phases. And it's moving towards the poles. I will show you when we get to that. So you need to differentiate between this terminology. Right. Now, this is a process of meiosis. If it's not clear with you there, but I'm going to give you just a second to observe what actually happens there, right? So, if you observe there, it was one cell initially, and it led to two cells. The other cell, it was one, it resulted into two cells, okay? What else do I observe there? Okay, let me get this part. It was a chromosome. Then at the end, each cell, each daughter cell had a chromatid. So this is the demonstration just to show you that when meiosis has taken place, we're going to result, it is going to result, or it will give us 4 different daughter cells. Can you see that? They end up being 4. And look at what we see inside chromatid. A chromosome at the end of meiosis. Each cell will be having a chromatid, right? We are going to look at the next phase. Remember, we have started with the preparation phase, and with the preparation phase, we're looking at the interphase. Now, after interphase, we have prophase, IP, remember? Prophase. Now, we've got prophase 1 and prophase 2. So what is going to help you again here is that you need to have all the cells drawn. Maybe you can use 2 papers. You draw meiosis 1 here, that is prophase 1, metaphase 1, anaphase 1, telophase 1. On the other page, you draw prophase 2, metaphase 2, anaphase 2, telophase 2. Then you should compare the 2 pages. You should compare all the phases, right? So that you are actually aware of the differences between the 2. Because in the exams, we can't be telling you that this is prophase 1, this is prophase 2. An examiner can give you can pick up just one diagram, and you should be able to identify the phase there and answer questions that will be given. Now, prophase 1, what are the features or what makes you to say this is prophase 1? You need to look at what is happening within the cell. Now, this is our prophase 1. The spindle fiber starts to form. Here are the spindle fiber. They start to form. And then homologous pairs of chromosomes. You can see homologous pairs of chromosomes. These are homologous pairs of chromosomes. Right? Now, if you compare prophase with interphase, remember in interphase you can only see the chromatin network. But In the prophase 1, they have formed what? Homologous chromosomes. You are able to see the chromosomes, and they are in their pairs. Hence, we call them homologous chromosomes. What happens in prophase 1 again? Remember, you can see with spindle fibers starting to form, and then chromosomes are arranged in the homologous pairs. What else? Crossing over occurs. Okay? Compare prophase 1 to prophase 2. If we are to compare the two, the nuclear membrane and nucleolus disintegrates. Right? You can't, the nucleus starts to disintegrate. Nuclear membrane starts to disintegrate. Then chromosomes have a section that is different. Let's check. These are the chromosomes where we can actually see the difference. Right? Now, here we just have homologous pairs, but then in prophase 2, chromosomes have section that is different. Now, where is that section? Can you see this section? Okay, with this one, I'll just talk about the color. This is gray, but this section is pink. Right? This is pink, but this section is gray. What makes that? So prophase 2 differs with prophase 1 because in there, nuclear membrane and nucleolus start to disintegrate. Chromosomes have a section that is different between the phases. Yeah, those sections are different compared to prophase 1. There is no crossing over here. But then in prophase 1, crossing over, when crossing over takes place, we know that like this one, we've got different sections. What do they mean to you? This means that crossing over causes what? Variation. But here, remember, in prophase 1, we only had homologous pairs of chromosomes, right? Right, when crossing over takes place, check this area is a point where crossing over is taking place, and this will lead to variation, or it causes what? Variation. So crossing over causes variation. Right. You can be given in an exam to explain what you understand by crossing over. Now, before crossing over takes place, chromosomes are in their homologous pairs. They are in their homologous pairs. That is before crossing over. Now, during crossing over, let's check. Remember, we've got maternal. This one will be from paternal. Now, during crossing over, what happens there? During crossing over, the chromatid of maternal overlaps with the chromatid of paternal. They are overlapping here. And the point where they are overlapping, we call it the point of chiasma or chiasmata. Now, remember, we have said for crossing over to take place, it indicates, it shows variation. It leads to variation. So crossing over has taken place here. Look at the next, that is after crossing over has taken place. What is going to happen? That's when we talk about those sections that are different. Those sections that are different. Right, the process takes place in prophase 1, okay? Crossing over takes place during prophase 1 of meiosis. And it involves exchange of genetic material between the sister chromatids, right? I've said to you, I will show you what sister chromatids are. So you can talk about sister chromatids where we see the genetic material being exchanged, right? Now, process of crossing over takes place in the prophase 1 of meiosis, involving what? Exchanging of genetic material. So at a point of chiasmata, that is where they overlap, the sister chromatids overlap. Then after overlapping, that's when we can see those sections where genetic shows that genetic material were exchanged. So it involves exchange of genetic material between sister chromatids, right? Sister chromatids of each homologous pair of chromosomes. So it is important to understand the process of crossing over. Think of what? Homologous pairs. What do? Sister chromatids overlap. After overlapping, you can see the exchanging of genetic material. So this section shows that the paternal exchanged this section with the paternal and maternal. They exchange these sections here. This means that genetic material were exchanged between the two sister chromatids of what? Of each homologous chromosome. Now, if you can check, I've been talking about homologous chromosome. So don't just refer to them as chromosomes because they are in pairs there. Right? Again, if we look at crossing over, when it has taken place, I have explained all of these stages here. Now, homologous pair of chromosomes are known as bivalents only when they are crossing over. Remember, we are talking about the homologous chromosomes. During crossing over, sister chromatids overlapped, right? Exchanging genetic material. Now look at this one. Homologous pair of chromosomes are known as bivalents only when they are crossing over. So we can talk about this as they are bivalents. Okay, they are bivalent. Why are we saying they are bivalent? It's because they are crossing over, right? All these states, they function as units or they connected to each other. Okay, now new maternal chromosome contains a piece of paternal chromosome and maternal chromosome. Hence we are saying after crossing over has taken place, genetic material are being exchanged because the chromatid from paternal overlaps with the chromatid of maternal, right? And immediately when we see that crossing over has taken place with those homologous chromosomes, then you can refer to them as bivalents, all right? So a new chromosome, this is a new chromosome. This is a new chromosome. Remember, this is before crossing over taken place. Then we've got a new chromosome. It contains a piece. This is a piece of paternal chromosome. This is a piece of maternal chromosome. So it looks easy, but then when you answer, make sure that you understand what is asked. Because yes, you know all this information, but should you get it wrong, it means probably you didn't read the question well. This is very, very simple. Right, stage after prophase, we've got metaphase. Meta means middle. All right, so with the metaphase, you are able to recognize the positioning of the chromosome to tell whether this is prophase, this is metaphase. Now on metaphase, also you need to compare the two phases. Metaphase 1 have metaphase 2. Before you can even read the questions, just look at what is happening there. How are the chromosomes arranged? Now, metaphase 1, pairs. We talk about pairs. That's very, very important. Pairs of chromosomes are aligning at the equator. At the equator, in the middle. This is the equator. Then we have random arrangement of chromosomes taking place. That is, they lead to source of variation. Compare metaphase 1 with metaphase 2. Now in metaphase 2, remember the main difference between metaphase 1 and metaphase 2. In metaphase 1, chromosomes are aligned at the equator in their pairs. But then in the metaphase, what is the difference? Chromosomes align singly. They are in pairs, as you can see. This is metaphase 1, but in metaphase 2, chromosomes are aligned at the equator singly, right? Then random arrangement of chromosomes takes place. I hope you can see the differences there, right? Now, please note that random arrangement of chromosomes, they cause variation. So I'm keeping on coming up with new terms. Now I've got variation. You need to understand what it means. All right, let's quickly summarize the metaphase 1 and metaphase 2. Chromosomes are arranged in their pairs at the equator, whereas in Anaphase 2, chromosomes are aligned singly at the equator. Then we've got random arrangement of chromosomes taking place in both. That leads to what? It causes sources of variation. Okay? Random arrangement causes variation to take place. Right. Now I'm going to stop here so that my colleague will do the anaphase part of it. Also, when you'll be looking at the anaphase, remember, throughout this meiosis, you need to compare meiosis 1 and meiosis 2. You need to compare the diagrams relating to the name of the phase, right? We've spoken about metaphase. Now it comes to anaphase. So if you have all those phases in front of you, looking at the arrangement of chromosomes, you can tell and determine the phase. Thank you so much. We are going to take a break, a short break, so that Samantha is going to finalize the last session. Thank you so much.
Good day, grade 12 learners. My name is Samantha Ragunanan. I'm going to take you through the second half of your section on meiosis. Just a reminder, you may contact us via WhatsApp on 079-454-9535. And a reminder again, no voice notes. You can send WhatsApp messages, but no pictures. Let's continue with Anaphase. So with regards to Anaphase, Anaphase is actually a very, very easy process to remember. It's also part of meiosis. It's also part of the whole reduction division process of meiosis. And it's if you look at the diagram, you would see that we've divided them into Anaphase 1 and Anaphase 2 simply because it's easier to study. And even when you are studying meiosis, you have to be drawing diagrams. You cannot be studying meiosis just by looking at the diagrams, because then you're not going to remember all of the different processes. So let us look at Anaphase. But before we go to the notes, let us just go to the diagrams and see what jumps out at us, because common exam questions on meiosis always have diagrams. You must be able to identify these diagrams, you must be able to label these diagrams, and you must also be able to discuss the processes, including all the organelles involved in the diagrams. So while you are busy taking notes as I speak, I've got some very easy tips for you to remember the different phases of meiosis. So with regard to Anaphase, Ana meaning apart, okay? I always try to remember Anaphase, they move away. Ana, away, okay? So what am I talking about? Here we know the labels here. So we can see in this diagram here, in Anaphase 1, we can see there is the equator. But what is happening? There's a space here. And whole chromosomes, double-threaded chromosomes, are moving towards opposite poles. You can see them. There they are. They're moving towards opposite poles. So that is how we know it's Anaphase 1. When we look at Anaphase 2, there's a very visible difference. If you look at Anaphase 2, there's still the space, nothing different there. But you can see it's a single-threaded chromosome. Can you see that? It is a single-threaded chromosome that is moving towards opposite poles. So in order for them to be able to move to opposite poles, something has to change. And this comes in with the spindle fibers. The spindle fibers actually contract. And when they contract, they pull them towards opposite poles. Okay? So let's look at it. So in Anaphase 1, the spindle fibers contract. Contract means they become shorter. When they become shorter, they move apart. They pull the chromosomes to opposite poles. It's like what we saw in the diagram. So sometimes, sometimes the words can sort of confuse us, but if you look at the diagrams, we'll be able to make sense. Oh, we can see the spindle fibers, they're getting shorter. We can see whole chromosomes, double-threaded chromosomes moving towards opposite poles. In Anaphase 2, yeah, the spindle fibers, there's the spindle fibers, it's contracting. That means it's getting shorter. And the centromere is split. So where is the centromere? There is the centromere. So in a normal chromosome, I know I'm great at drawing, not, but what happens here is that the centromere, there's the centromere, okay? What happens is that it splits and then they become, they separate the two chromatids and they become single threads. And that is how we can see in this diagram that they're moving to opposite pole. So that's what happens during Anaphase, okay? If we move on to telophase or telophase. So I always remember telo 2, T for 2, isn't it? So telophase means 2. So what happens between telophase 1 and telophase 2? Now very, very simply, we're going to do the same thing. Even when we're studying, we're going to do the same thing. We're going to look at the diagram, we're going to try and label the diagram, and from the diagram, we are going to try and make sense. Now already I can see something happening here. I can see a C-shaped structure, okay? And I can see an invagination occurring or a constriction occurring here. I can also see that there is a nuclear membrane there. There's 2 nuclear membranes, and I can see that that the cell is like starting to divide into 2. One cell is going to be dividing into 2. So let's see what it says. The nuclear membrane reforms. The cell membrane constricts. There we saw it. C4 constricts. And cytokinesis occurs. Now, what is cytokinesis? It's just a fancy word for saying that the cytoplasm splits. Okay? So the cytoplasm divides into 2. But before it divides into 2, whatever is in the cell, it doubles itself. And the nucleus also splits and it divides, it doubles itself. It makes double copies. So they move to opposite ends of the cell. So why is this important? This is important because to make sure that the proper chromosome, the correct chromosome number is in the daughter cells. Okay? So here you have double-threaded chromosomes, consist of 2 chromatids and a centromere. There's the 2 chromatids, there's the centromere. And in telophase 2, it looks a little different. Can you see the difference between telophase 1 and telophase 1? Here we have double-threaded chromosomes. Here we have single-threaded chromosomes. Here you can already see the centromere has split and it is now single-threaded. So that is how. So if you want to know how to distinguish between meiosis 1 and meiosis 2, if you look at meiosis 1 in telophase, there are whole chromosomes, double-threaded chromosomes. But if you look at telophase 2, they are single-threaded chromosomes, because sometimes we become confused. We know it's telophase, but we don't know which one. And the question becomes confusing. That is why it's important when you get a question, look at the diagram first. Like my colleague Agnes has alluded to, you need to read the question very, very carefully before you proceed with the question, proceed to answer it. So it's single-threaded chromosomes, big difference between telophase 1 and telophase 2. Each daughter cell is half the number of chromosomes, and it can be half of the number of chromosomes. Why? Because cytokinesis has occurred. And 4 haploid daughter cells are genetically different. So here we're going to have, there's the first one, it's going to be haploid. The second one is going to be haploid. Number 3 is going to be haploid. And number 4 is going to be haploid. There's a visible distinction between the 4 haploid daughter cells that are formed. Okay, very, very easy, very simple to remember. If you can remember the difference between meiosis 1 and meiosis 2 and the phases where they differ, it can help you a lot. So you can do this even on a double page, and you can draw meiosis 1 on the one side, for example, telophase 1 and telophase 2, and you can put it up in your rooms, on your walls. I promise you, you won't have nightmares, but it will just help you to remember that much better. Okay, now let's talk about meiosis versus mitosis. So we know that in our grade 12 exam guideline, because that is our point of departure, we know that we don't focus too much on mitosis. The last time we heard about mitosis actually was in grade 10. Okay, so we're not going to focus too much on mitosis. We're going to focus on the process of meiosis. But you need to be able to compare the two, because they like to ask that. Because remember, your grade 12 work is a culmination of grade 10 and 11. So this is relevant previous knowledge which you're supposed to know. Okay? But just to refresh your memory, I'm going to discuss meiosis versus mitosis. So let's talk about meiosis first, because that is what our lesson is on today. So if you look at the definition of meiosis, it makes distinct gametes. And it changes from diploid to haploid. Now, what does that mean? It makes distinct gametes. So the whole point of meiosis is to make gametes. For example, the ovum and the sperm cell. Okay, we need gametes because we need to move from a diploid cell to a haploid, because when they combine, there is no doubling of fertilization. Okay, so let's look at what is going on here. So when it say they make distinct gametes, I want you to look at this diagram. Look at this diagram, look at that diagram, and look at the third diagram. If you look at all the chromosomes in each of these cells, they differ. Look at the bottom line. None of these cells look identical. What does it tell us? It tells us that they make distinct gametes. What does distinct gametes mean? Distinct means they are visibly different. That means we can see them with our eyes. We can see that they are different. That one's got a little bit of red. That one has got all red. So we can see that Listen, this is what meiosis is all about. It's the formation of these gametes. And the main thing you must remember about meiosis is that it's a reduction division. What is reduction division in simple terms? It means it changes a cell from diploid to 4 haploid cells. And the resulting cells are different from the original cell. So meiosis is important because what it does, it helps us to adapt to our environment in everyday life. So it also gives us genetic difference. We look similar to our parents, but let's face it, we don't really want to look exactly like our parents. So that is a great part about genetic difference, is that you have those genes from your parents, but because the process of meiosis, maybe you have more genes from your mother, less from your father, and that is why people will say, oh, you got your mother's nose. That is all part of the process of genetic variation, okay? Now let's look at mitosis. How is it different? Now, firstly, it's different because it starts off from diploid and it moves to diploid. So it starts off as 2N and it ends off as 2N, very different from meiosis. So mitosis is basically the cloning of body cells. Sometimes you wish, I wish I had a clone. I could send this clone to school and I can sit at home and watch Netflix or go on Instagram, but no such luck, okay? So that is what mitosis actually does. It makes a replica. It's a multiplication process where it multiplies the body cells, okay? So this is a reduction division because 2N to N, and this one is the same ploidy, which is diploid and diploid. So mitosis is very like meiosis too. You'll see what I mean just now. Let's look at the comparison between meiosis 1 and meiosis 2. Now remember, Agnes has been through prophase 1, prophase 2 with you. Then it is metaphase, then anaphase, and telophase I went through with you. So now we need to compare. How is meiosis 1 different? Why can't it just be the same thing? It's not the same thing. Let us see why. In meiosis 1, chromosomes arrange at the equator in homologous pairs. I want you, while I'm talking about the comparison, I want you to guess which phase of meiosis it is, okay? Chromosomes arrange along the equator. The equator is the middle, remember? Meta is middle. And in meiosis 2, chromosomes line up at the equator individually. Okay, so do you see what they're doing here? They're comparing meiosis, metaphase 1 and metaphase 2. Because in metaphase 1, they arrange themselves in homologous pairs. That means there's groups of the chromosomes. Here, the chromosomes, they arrange themselves individually. What is the difference between meiosis 1 and meiosis 2 when it comes to the next one? Whole chromosomes are pooled. The moment I hear the word pooled, I think they're moving away, apart. Anaphase, you remember that? Let's see. Whole chromosomes, so it's whole chromosomes are pulled to opposite poles of the cell by the spindle fibers. Remember we spoke about the fact that spindle fibers, they contract or they become shorter. And when they become shorter, what do? They pull the chromosomes to opposite poles. Here, in meiosis 2, chromatids are pulled to the opposite. Chromatids. Do you see it's chromatids? It's a single thread. So it's chromatids which is pulled to the opposite poles of the cell. So we can already see that is anaphase 1 and that is anaphase 2. Okay, because we know our content now. So what we need to do is we need to apply to the question which we are given. Let's look at the third comparison. So so there's a trick here. Do you see what's happening here? When the examiner asks you, compare meiosis 1 to meiosis 2, all you have to do is remember all of the phases of each one. Prophase, metaphase, anaphase, telophase, and compare the two. And that's how you get your table. So what was in diagrams, you are now putting it into words, okay? So there are 2 cells formed at the end. Remember 2, telo 2, isn't it? So that would be telophase 1. And 2 cells are formed at the end of this division. Remember the cytokinesis occurs and all of that? They're talking about this, but they're summarizing it. And then in meiosis 2, 4 cells are formed. I'm not sure if you are actually picking this up, but meiosis 2 is almost exactly like mitosis. So that is one of the things that I use when I'm answering exam questions. I look to see if there are double thread chromosomes or homologous chromosomes. The minute I see homologous chromosomes, I know, hey, this must be meiosis 1. And if I see single thread chromosomes, and obviously this will be from anaphase onwards, I would know that this is now meiosis 2. Okay? But as you can see, this meiosis 2 is very different from meiosis 1 because crossing over does not occur. And we know that crossing over occurs in meiosis 1. Remember, it takes place in prophase 1. So crossing over occurs. When crossing over occurs, there's genetic variation. So that makes the daughter cell different from the parent cell. But remember the cloning, the mitosis, they are exactly the same. They look the same. And that is very similar to meiosis 2. Okay? So the reduction takes place mostly in anaphase, from anaphase onwards. The chromosome number is halved during meiosis. Very, very important observation. That's the main thing, because the whole point here is we want to make gametes. For example, we need sperm cells, and those sperm cells need to be haploid. So that haploid sperm cell and the haploid ovum can form a diploid zygote. Or else, the zygote is going to have not 10 fingers, 20 fingers. 3 eyes, 4 eyes. No, we can't have that. We'll be looking like aliens. Okay? So we need this process of meiosis so that we can look normal and look great and carry on with life and adapt to our environment. Now, meiosis 2, the chromosome number remains the same. Wasn't this the case in mitosis as well? So meiosis 2 and mitosis is very, very similar. Okay? So this is now how you would compare. You would name all the phases, go through all of the phases, compare. Okay? A comparison question. So you'll get marks for this. 1, 2, 3, 4, 5. Plus you'll get an extra mark for actually drawing the table. So you need to draw the table, which must be T. You must have the heading, the main heading. You must have the subheadings. And the fact that it's in a table, you will get marks for that. So that is how this applies to different tables that you would draw in the matric exam. Okay? So if we move on, let's get to the fun part, because now we learn all the content. We know the content. So let's see if we really know the content. And this is what you need to do, grade 12. You always have to test yourself, because you think you know something until you come across a question. And they like to ask similar style of questions, but in different ways. So you can put yourself at an advantage by going through the questions so you have an idea. It will sharpen your question answer skills. Okay? Let's see. Now, I'm not going to look at the questions. You know me. I always look at the diagram first. I make sense of the diagram, and then I go to the questions. So here in the first diagram, okay, already at A, I can see that crossing over has occurred. How can I, because it's patchy, isn't it? Here I can see there's a process of crossing over. Can you see that? There is a process of crossing over, and these are different stages of the cell. So I'm looking, there's a label there. The fact that it is crossing over, obviously when they cross over, the point at which they cross over is the chiasma, isn't it? It's the point at which they cross over. And then the mother cell or the maternal part of the chromosome breaks off and it joins to the paternal chromosome, and the vice versa for the paternal chromosome. That is how they exchange genetic material. Okay, but already when we know it's crossing over, it cannot be any other phase but prophase 1, because crossing over only takes place in prophase 1. Don't let them trick you in the exam. Know where it takes place. So let's continue. How many pairs of chromosomes occur in a normal human cell? Now already my first instinct is to say 46, because we learned this, isn't it? Human is made up of 46 chromosomes. But they're asking how many pairs. How many pairs make up 46? And that would be 23. So those of you who are saying 46 and shouting out 46, you need to read the question correctly. Look for keywords. Give the labels of structure X. So what is X? We all know this. This is the centromere. Now remember, centromere and centriole are two different structures. Centromere is what joins the two chromatids together, and the centriole is found at the poles of the cell. So let's look at structure X. Let's see if we are correct. Yes, it's centromere. Area Y. Remember, the area in which they cross over, we said, is the chiasma or singular chiasmata. Okay? Name the organ in the human female where meiosis occurs. Now, they're asking keywords. Organ, it's a female. So we know there's nowhere, sperms is nowhere here. And it's meiosis. What did they say is the purpose of meiosis? It's reduction, division, and it's there to form gametes. Where is the gametes formed in the female? In the ovary, correct. It is formed in the ovary. So you need to break up the question so that you understand what they actually need. Same diagram, more questions. Name the process occurring at diagram B. We all know this. If you do not know this, you have not been paying attention at all. What is this process? It is called crossing over. What is crossing over? The exchange of genetic material. What phase is represented by the diagrams above? Now remember, crossing over, I said over and over again, crossing over only takes place in prophase 1. Hence, the answer will be prophase 1. Okay? Remember in prophase 2, the chromosomes already had their patchy appearance. Crossing over has already occurred. So we can't call that crossing over. The type of cells that would result from the meiosis of the cell. What type of cells would result from the meiosis of the cell? What type of cells are we looking at? We're looking at obviously gametes. Remember, we said the whole aim of meiosis is to form gametes. So we can have haploid structures, okay, for continuation of generations. So that was simple enough, okay. This was focusing, and they like to ask, common exam questions is talking about prophase 1. So if you want to start studying meiosis, a good point to start is with the crossing over, the labeling of the diagram of crossing over, and knowing which phase it is in meiosis 1, which is prophase 1, okay. Let's move to this diagram. So now we know that things are going to become a little bit more difficult, but we're going to get a variety of questions. And mind you, all of these questions are from past year papers. So it's not like I'm thinking up all of these questions. They're from past year papers, and you should know how to answer every single one of them. Okay? Some of them are from last year's paper as well. The diagram shows the somatic cells. Somatic. Now I want you to get used to this word. You must know what it means. What is somatic? It is body cells. It is body cells. And what do we know about our body cells? Are they sex cells? No, they're not. So that what would it mean? That they are diploid. They always will be diploid. So they are not, they are very unlike the sperm cell and the ovum. They are diploid. Okay? Because they make up our body. So that is what the question is saying. So the diagram shows the somatic cells of an organism in one of the phases of meiosis. So they're not telling us which phase this is, but we have a diagram, and they're saying it's a phase of meiosis. And we can already see that they want labels of the centromere. They want some information about this diagram. And the fact that I'm looking at this diagram now, and the first thing I'm asking myself is it meiosis 1 or meiosis 2? How do I know? I know because I'm looking, I can see. Can you see these chromosomes, these double thread chromosomes? They have not split yet. They have not separated. So that is a clue for me, because I know I'm not dealing with meiosis 2 here. But let's wait and see, okay? Give the letter and the name of the part that carries genetic information. What carries genes? What carries genetic information? You did this last week. What would that be? It would be B. Oops. So it carries genetic information. But already I'm looking at B here, and I'm already saying, okay, it's chromosome. That's what I did initially. When I looked at the question, I said it's chromosome. But I'm wrong. I'm half wrong, because I'm only going to get 1 mark. Because I'm saying chromosome. They're not asking for just chromosome. They're asking the letter and the name. And it's 2 marks. Grade 12s, you have to be very, very careful. Very, very careful in how you're answering. Because do you see how even if you know the content, you won't get full marks if you don't read the question. Don't be stressed out during the exam. Be calm, read, underline keywords, and take your time. Okay? You are given 1 minute for each mark. It carries genetic information, so I'm going to say B and I'm going to say chromosome. Okay? You see where the tick is B, chromosome, then I can get the 2 marks. What holds the chromatids together? What holds the chromatids? We know this. We've mentioned it over and over. It will be E because I'm giving the letter first, and it's going to say centromere. If I continue, let's look at the number of chromosomes, same diagram. State the number of chromosomes present in each of these cells of this organism. Now they're saying this organism. Initially, you will always assume that it's a human. You can't assume. They're talking about this organism here. So you're going to only have to be talking about this organism at the beginning of meiosis. So because we can see that the homologous pairs have not split yet, they have not separated, we have to count the number of chromosomes here. It's 1, 2, 3, 4 because it's at the beginning. Remember, in the beginning of meiosis, you have the full set of chromosomes. After meiosis, after the separation is done, you have half the number. So at the beginning of meiosis, we are going to have 4. 1, 2, 3, 4. And then they're trying to trick you here because they're saying in a gamete of this organism. And we know what happens after meiosis, gametes are formed. So this is they're talking, the beginning is before and the gamete is after. So if it was 4 in the beginning and it is halved, it's obviously going to be 2. Do you see? Because normally most learners would assume that it's a human's somatic cell and they'll just say 46 chromosomes and then it's 23 gametes. You can't, okay? You must look at the question and see what clues they are giving you. So let's look at this one. This is a different sort of diagram. And the diagram below shows cell P in a phase of meiosis. Do you see how tricky they are? They hardly ever tell you which phase it is. But every question is about identifying the phase. Name the phase of meiosis represented in the diagram. So what can I see from this diagram? Okay, let's see. If you look at this diagram here, we can see crossing over has already occurred. Because do you see that the appearance of the chromosomes? So prophase 1 is gone. So it has to be after prophase 1, okay? And do you see here, so the chromosomes, the chromosomes are aligned in the middle. Remember, the middle is metaphase, middle meta, okay? So I'm thinking it could be metaphase because they are aligned in the middle. But I remember there was a distinguishing characteristic between metaphase 1 and metaphase 2. In metaphase 1, the chromosomes were arranged in pairs at the equator. They were arranged in pairs. But in metaphase 2, they were arranged singly at the equator. So what would you say is the name of this phase? And you are correct in saying it's metaphase 2. It can't be metaphase 1 because then we're going to have diagrams of chromosomes, homologous chromosomes next to each other. So you are correct in saying that. Give the number of chromosomes that were present in the parent cell of cell P. Now, look at your previous question. It says metaphase 2. So what does this mean? Separation has occurred, isn't it? Because some of the homologous chromosomes went to one side of the pole, the others went to the other side of the pole. So when we look at this, the parent cell would obviously, it would be 4 because this is meiosis 2. In meiosis 2, if there are 2 left, what would it mean? It was halved. So if it's halved, the original would be doubled, which is 4. And one daughter cell at the end of meiosis. The daughter cell, it's almost always half of the original, which is 2, as you can see. Okay? So there's it, 1, 2. We actually just have to count because it's meiosis 2. Let's move on. Same diagram. We are rounding up this diagram now. The diagram below shows cell P in a phase of meiosis. We already know it's metaphase 2 because we established that. Give the name of the structure. So they want the name of the structure, and we can see it's a chromosome, okay? And then they want this. And last year's paper, they concentrated on the spindle fiber. So you need to know what is the function of the spindle fiber. Obviously, B would be the spindle fiber. And what it does is we need to know a spindle fiber, it pulls chromatids or chromosomes to opposite poles, not just any poles, because a spindle fiber can pull it to the same pole. It can pull all of this to the same pole. No, we are saying that the spindle fiber shortens and it pulls the chromosomes to opposite poles. So this definition, if I were you, I would learn this definition of what is a spindle fiber and how it functions. When it contracts, what happens? Okay? If we move on, activity 3. Describe the process of crossing over that has resulted in the appearance of structure. So my colleague has covered this because we know what crossing over is. Crossing over, let's think about it before we look at the answer. Crossing over takes place, we must name the phase. Where does it take place? It takes place in prophase 1. The second thing, homologous chromosomes, they come to lie next to each other. It's obvious. Grade 12s, they can't be far apart and then be crossing over. They come to, that's a clue. They come to lie next to each other, and what happens? The one chromatid of the other, they cross over at a point called the chiasma. And part of the neighboring chromatid becomes part of the homologous chromosome that is adjacent to it, and they become, the genetic material is exchanged. Okay? So let us see if we've described the process in our own terms, because it's not enough to know the diagram. It's not. They ask you to explain what is going on. So there they said during prophase 1, you see I did not lie. It's prophase 1. Homologous chromosomes lie next to each other. They have to lie next to each other. Chromatids from each homologous chromosome are non-sister. They're non-sister because they haven't exchanged yet. They overlap. They overlap, remember the diagram? And then they touch each other. It's obvious. You are stating the obvious. In order to overlap, to exchange, they must touch each other at a point called the chiasma. And the chromatids exchange genetic material. So this is 4 marks. The tick is on prophase. It's on homologous chromosomes lying next to each other. It's on overlapping. And it's on the chiasma. And if you have exchanged genetic material, you will also get a mark for that. Okay? So remember, you need to know where crossing over takes place. You need to know the process of it. Very, very important. It's a common exam question. Right. Now, do you see the variety? If you look at this graph, you're thinking, how could they possibly fit meiosis into a graph? They have, and they've done it, and they're still going to do it. So if you do not know how to draw graphs, you don't know how to interpret graphs, it is a skill that is needed for life sciences. Okay? Let's read the question, and then before we go to the question, we're going to try and interpret the graph. So, the number of chromosomes in somatic cells. The minute you see somatic, grade 12s, please remember, somatic cells are diploid. You have to remember that because that is a clue they're giving you in the question. They body cells of organisms differs from species to species. We know that. The graph below shows the number of chromosomes in each somatic cell of 3 species. So do you see that each species, it's all, we as humans, we've got 46 chromosomes, but not all organisms have 46 chromosomes. Let's see. The giraffe has 30 chromosomes. I read it off the graph. You are not going to read it off the graph, grade 12s. You are going to be much better than I am. You're going to have a ruler, and you're going to take your ruler and you're going to check it, because this is for marks. This is your future you're dealing with. You can't afford to take chances. The mouse has 40 chromosomes. And the pineapple plant has 50 chromosomes. So I already know this. But remember, it's somatic cells. So if I have to look at sex cells of any of these, is it going to be the same number? No, it won't, because these are diploid. If they undergo meiosis and we need sex cells or we need gametes, we need them to be haploid. So it will be half this value. Let's see what the question asks. How many chromosomes will be present in the mouse cells during telophase 2? Now, the fact that they're saying telophase 2. It's flagged to me because in telophase 2, the gametes have already been formed. So the mouse cells, if I look at this, it says 40. But telophase 2 has the gametes are formed. So it can't be 40. It has to be half of 40. And that would be 20. Do you see how important it is to read the question carefully? A leaf cell of the pineapple plant. How many chromosomes? Okay, here's the pineapple plant. Okay, but they're talking about the leaf. Now, when we talk about the leaf, they could have just said the cell of a pineapple plant. But the fact that they said leaf, it means they're giving us a clue. Because we know from our grade 10 and 11 work that a leaf, the function of a leaf, when we were in primary school, they used to say it's a food-making organ, okay? But when we came to high school, we said it's where photosynthesis takes place. So if it's involved in photosynthesis and carbon dioxide oxygen exchange, it is not involved in reproduction. If it is not involved in reproduction, the chromosome number will be the same because remember, they are somatic cells. So it would remain as 50. Do you see how they can trick you? So you need to pay attention. Explain why the sperm cell, already I see sperm cell, in my mind it's saying gametes, 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 okay? So the sperm cell I already know it must be haploid, okay? Explain why the sperm cell of the giraffe has 15 chromosomes, and they're spot on because all the chromosomes of the giraffe has 30, but the sperm cell has 15 chromosomes. So they want to know why. Why? Because the sperm cell, firstly, it's haploid and it is a gamete, so it must have half the number of the total set of chromosomes, and it's also to prevent doubling of chromosomes during the fertilization process. So let's see if they have the same idea that we do. A sperm cell is a gamete, very important. It is formed by meiosis because meiosis must occur for that gamete to be formed, and it must be haploid. So it's first meiosis, then it's haploid, then we have a gamete. And to overcome the doubling effect of fertilization. So if we have haploid and haploid, we can get diploid. But if we have diploid and diploid, we've got problems because we have extra organs and it's just going to be a big mess. So we have to have meiosis taking place. Very, very important. Okay, let's look at the next question. Name the phase of meiosis where the halving of the chromosome number begins. So the halving of the chromosome number, we will remember that when they move away and they're halved, it would be anaphase, isn't it? So it's anaphase 1. Describe the events of the phase mentioned in the previous question, number 3. Anaphase, remember we said the spindle fiber shorten, the chromosomes, the whole chromosomes are pulled towards opposite poles, and the chromosomes pairs separate, and that is going to be for 3 marks, okay? And then the next question, the diagram alongside represents 2 phases of meiosis. So if you look at these 2 phases, I can already see there's a space here, so I know this must be anaphase something. I can see all chromosomes, and I can see that crossing over has occurred, okay? So crossing over has occurred, but because it's whole chromosomes, I would guess that it's anaphase 1. Here I can see single thread chromosomes. I can see invagination occurring, and I can see that they are moving apart. So I would take this as telophase 2 because of the single thread chromosomes. Let's see, identify part A. It is not the centromere. The centromere is what joins the chromatids together. It is the centriole or the centrosome. Identify the phase. They're talking about diagram 1, and that would be anaphase 1. We know why, because it's after prophase crossing over has occurred. Name the process that causes the chromosomes to have a combination of genes in these diagrams. You know this, crossing over. We've went over it, okay? Give one reason why the process named is important. We know why. Remember we spoke about your parents? We said that we need to have genetic variation. That means we all can't look the same, okay? if this was a human cell, how many chromosomes would be present in diagram 1? Now remember, diagram 1 represents meiosis 1. So how many chromosomes are there before the gametes are formed? And that would be 46 chromosomes and 23 pairs. Structure B and C are both chromosomes. Explain why they are structurally different. So we need to look at them. B and C. One is double-threaded, the other is single-threaded. We know that here we're going to have 2 sets of DNA because there's 2 chromosomes. Because this is a result of DNA replication. And here we have a single molecule of DNA. So that would be the difference between the two. Structure B consists of 2 DNA molecules. Because of DNA replication, structure C consists of 1 DNA molecule because it is unreplicated or single-threaded. Okay? The figure alongside represents a cell during meiosis 1. Here they were kind. I like this examiner. They actually gave us a clue. They said it's meiosis 1. Identify the stage of meiosis. Now, I can see that crossing over has occurred, so it's after prophase 1. I can see that we have homologous chromosomes that are lying next to each other. They're in pairs. So I know this has to be meiosis 1. And because they are in the middle or at the equator, I know that this must be metaphase. And it can't be metaphase 2 because metaphase 2, the chromosomes, the homologous chromosomes are singly arranged. That means it won't have this layer, it would only have 1. But because they are double, it must be metaphase 1. Very good. Okay, if you guessed that, very good. Give a visible reason, exactly what we said. It is at the equator and we have homologous pairs lying next to each other. There's the homologous pairs and there's the equator. Keywords when you are answering. Provide labels for structure 1 and 2 respectively. Structure 1 would be the chromosomes, isn't it? The homologous pairs which we've just identified. And structure 2, they are not pointing at the whole chromosome. They are pointing at individual single threads. So because they are single threads, we call them sister chromatids. Okay? And then, would you consider the cell shown in the figure to be haploid or diploid? I would consider it because I can see homologous. Remember I told you the clues we need to look for. Crossing over has occurred, but nothing has been, these chromosomes have not been split as yet. They have not been separated. So I would consider them to be diploid because there is no, meiosis has not, the process has not been completed as yet. So yes, it is diploid because we see homologous chromosomes. And because they have not separated. See, the centromere is still intact with both chromatids. Okay? Homologous pairs have not been separated and they are still present. See, it becomes easy as we go along. How many chromosomes would be present in each of the daughter cells from this cell at the end of meiosis? So this is before the starting of meiosis, and at the end of, so the starting of meiosis, we got 1, 2, 3, 4. So at the end of meiosis, in meiosis 2, we're going to have only 2 because it is a reduction division and the ploidy is halved. Okay, so grade 12, that brings us to the end of our presentation, and I hope that you have taken from this lesson the variety of ways in which grade 12s can be asked on the topic of meiosis. It's not just a matter of knowing the diagrams, it's a matter of knowing the difference between meiosis 1 and meiosis 2. And the different phases in each one. And remember, crossing over takes place in prophase 1. Okay, so good luck with your control test, and we will see you next week for the second half of meiosis. Thank you.